Hello everyone and welcome to the complete course on basics of plant breeding. Myself, Dr. Narkade Gopal Vasudev, working as Assistant Professor, Department of Genetics and Plant Breeding, School of Agriculture, SR University, Warangal. And I'll be your instructor throughout this course. So, I have divided this complete course on basics of plant breeding into total 20 lectures, which will start from Introduction to Plant Breeding till Intellectual Property Rights in Agriculture. Right? So here we will see a quick overview on different topics, what actually we will cover. So as I told you, lecture 1 is Introduction to Plant Breeding. So in this lecture, we will going to see the definition and scope of plant breeding, which will deal with improvement of plant genetic makeup for human benefit. So next and most important point is concept of genetic improvement. So in this concept of genetic improvement, we will see selection and recombination of desirable traits. The third point is nature, art and science that combines the scientific principles with the breeder skills. Our next point is role in agriculture. What is the role of plant breeding in agriculture and food security? So, plant breeding will enhances the yield, quality, and resilience. Then next we will going to see what are the aims and objectives. So, like development of superior, stable, and adaptable varieties for different crop species. And last but not the least, importance in modern agriculture. So like plant breeding will support the sustainable and climatic resilient farming. So these all the points we will cover in the lecture 1, that is introduction to plant breeding. So in next lecture, that is lecture number 2, we will see the historical development. What the historical developments are there in plant breeding. So right from first point, that is origin of plant breeding, that it begin with early agricultural practices and selection. So second point in this lecture two, like domestication as a first breeding method. So like conversion of wild plants into cultivated forms. Third point, that is early hybridization work, that is first artificial crosses improve with improved crop traits. Next, that is rediscovery of Mendel's laws in 1900, which laid the foundation of scientific plant breeding. For this purpose, Mendel regarded as the father of genetics. The next is development of modern techniques like integration of genetics and biotechnology with plant breeding. And again, last but not least, that is future prospects, that is focus on precision breeding and climate adaptation. So these are some points which we will cover during lecture 2, that is historical depth. So our next lecture, that is lecture 3, which includes genetics in relation to plant breeding. So in that first we will see what is the role of genetics in plant breeding. Here we will see the inheritance and variation in crop plants. Then after that we will see the Mendelian laws, which will govern the segregation and assortment of genes. Then next we will see the dominance and gene action. So this dominance and gene action which will determine the expression of the traits. Then quantitative genetics, like this quantitative genetics means study of polygenic traits which are having the complex inheritance. So for example, it is yield of a particular crop. So again, one most important point for quantitative genetics is heritability and genetic advance. which will predict the response of that particular trait to selection. And last point in this lecture 3 is application of genetics in plant breeding. So like genetics will guide efficient crop improvement strategies depend on heritability and genetic advance. So that's why the heritability and genetic advance, these two are the major parameter or major factors considered for crop improvement. Then from genetics in plant breeding, then we will move towards the lecture 4, that is modes of reproduction. So as everyone well aware, like there are two modes, that is sexual and asexual. So sexual reproduction, it involves the fusion of male and female gametes. And in asexual reproduction, this asexual reproduction produces the genetically identical offsprings. The next point is natural versus artificial propagation. So this occurs naturally or through human intervention. Then in this next topic in this modes of reproduction, there is concept of apomixis, like formation of seed without fertilization. Next point, that is types of apomixis. Like it includes the parthenogenesis, apogamy, and apospory. Then last point, significance of all these modes of reproduction in plant breeding. So like it helps to fix the desirable traits permanently. So from modes of reproduction, we'll move towards our next lecture, that is most important, male sterility. So in this first we'll see what is exactly the male sterility, that is failure to produce the functional pollen. Then next we'll see what are the different types of male sterility. So like there are three types, that is genetic male sterility, cytoplasmic male sterility, and cytoplasmic genetic male sterility. So these three types we will see. Then the next point is mechanisms of sterility, that is caused by genetic or cytoplasmic and genetic factors. What is the role of male sterility in hybrid seed production? And simultaneously, we will see what are the different advantages. Why male sterility is preferred in hybrid seed production or in plant breeding. Why male sterility is preferred? Why researchers are preferring male sterility? Like it will reduce the labor and increase the hybrid efficiency. So ultimately, if hybrid efficiency will increase means ultimately you will get the higher percentage of heterosis. So then from male sterility, then our next lecture, that is lecture number 6, that is plant genetic resources. So in this plant genetic resources, we will see the what is meant by germplasm. So this germplasm is the total genetic material of a species. Then next, what are the types of plant genetic resources? Like it will include the land races, wild relatives, and cultivars. Then importance of this plant genetic resources in crop improvement. That is sources of useful genes. We can find the various useful genes. Then what are the conservation methods like in situ and ex situ preservation of germplasm? Then what is gene bank? Like it will store and maintain the genetic diversity for particular crop species. And then last point, whatever this germplasm, plant genetic resources, conservation methods and gene banks, how we can utilize all these things in plant breeding. Right. So that thing we will see. So our seventh lecture will deal with domestication of crops. Like first we'll see what is mean by domestication. So what is domestication? It is bringing wild plants under cultivation. What is the process and what are the different stages of domestication, like selection, adaptation, and cultivation? So, what are the different genetic changes, like loss of wild traits and gain of useful trait? That is the most important genetic change occurs during domestication. Then next point we will see, that is role of selection. So with all this, a breeder or a human can select the desirable characteristics. Then we will see different centers of domestication, that is regions where crops originated. Then what is the importance of domestication of crops in crop improvement? So this all these things we will see in lecture number 7. So next lecture number 8, that is introduction. That is introduction of germplasm. So in that first we will see the definition. That is what is actually introduction means what? That is transfer of crops to completely new environment. Okay. Then next, what are the different types of introduction? That is primary and secondary. That also we will see. Then direct versus indirect introduction. So means you are introducing particular crop for immediate use or for further improvement. So on that basis also you can classify like direct introduction and indirect introduction. What are the objectives of this introduction program? So like the basic objective is to improve the yield and adaptability. Then next we will see acclimatization. What is the meaning of acclimatization? So this acclimatization means adjustment to the new climatic condition. So like first we will see introduction means transfer of new crops to totally new environment. That is introduction. And adjustment to that new climatic condition, that is called as acclimatization. And at last we will see what is the role of this introduction and acclimatization in plant breeding. So next lecture is center of origin and center of diversity. So in this lecture, first we will see the concept of origin, that is place where crop first evolved. Then next we will see the Vavilov state, that is centers with high diversity are the origins. Then next point, primary and secondary center, that is original versus diversification regions. What is the importance of this center of origin and diversity in plant breeding? Then next point we'll see, that is identification methods, that is based on diversity and based on evidence. Last point in this lecture we will see role in diversity. What is the role of this center of origin in maintaining the genetic variability of the particular crop? Ultimately, this total plant breeding is dependent on this genetic variability. So, this maintenance of genetic variability is the critical step. Okay. So, this 10th lecture, pre-breeding. So, like generally we'll see what is breeding means. Everyone will tell like crossing between two parents to get hybrid. But to develop the superior parents for development of hybrid, that is most important thing. That process is called as pre-breeding. So like transfer of useful genes from wild species into cultivated one. This pre-breeding is also called as germplasm enhancement. Right. So what then next we will see what is the role of wild relatives in pre-breeding program. What are the different techniques we can use? That is hybridization and gene introgression. And what is the importance of pre-breeding? That is in creating new variability. And how we can link this pre-breeding with actual breeding program. So that also we will see in this lecture number 10. So this pre-breeding is most important topic. Then in lecture number 11, we will see the different breeding methods for self-pollinated crop. So as we know, self-pollinated crops are highly homozygous and uniform. So there are different methods, like first is pure line selection. That is selection from a single plant. Then next is method is mass selection. That is selection based on phenotype. Next you will see pedigree method. That is selection with a record of ancestry. As the name itself indicates, pedigree. Next method, that is bulk method. Selection after several generations. And last but most important breeding method is backcross breeding. That is transfer of trait from donor to recipient parent. So this all breeding methods we will see in lecture number 11 with respect to self-pollinated crop. So in lecture number 12, after self-pollinated crop, we will move towards cross-pollinated one. So this cross-pollinated crops are highly heterozygous and you will find lot of variation. Right. So next we will see population improvement for enhancing the gene frequency. So as far as the breeding methods are concerned, we will see mass selection. So in this selection will be there from open pollinated population. The next breeding method in cross-pollinated crop species is recurrent selection, that is repeated selection cycles. Then development of synthetic varieties, that is this synthetic variety is developed by selected genotypes. And here last but most important, that is heterosis breeding. So it will exploit the hybrid vigor. Okay. So lecture number 13 itself it is on heterosis and inbreeding depression. So first we will see what is meant by heterosis. So heterosis is superiority of F1 hybrids over the parents. Like hybrid vigor, this heterosis is also called as hybrid vigor. So what is hybrid vigor? It is increased yield and performance. So the genetic basis of heterosis, next point. So in this genetic basis, we will see dominance and overdominance effect. So next point, inbreeding depression, that is loss of vigor due to homozygosity. Then what is the role of heterozygosity? That also we will see. And application of this heterosis breeding in hybrid seed production. So this is all about lecture number 13. We will see heterosis and inbreeding depression. So in lecture number 14, we will see the development of inbreed lines and hybrids. So these inbreed lines are highly homozygous. Development of inbreed lines achieved through repeated selfing. Then after development, we need to evaluate. So testing for performance and stability. So that means you need to evaluate the inbreed lines multi-location. Then next we will see the combining ability, that is ability to produce superior hybrids. And how you will develop hybrids? Whatever the inbreed lines you will select, so crossing between the selected inbreeds. And what is the importance? So it will improve the crop yield significantly. So all these things we will see in lecture number 14, that is inbreed lines and hybrids. So this lecture number 15, it will depend on asexually propagated crop. Like asexually propagated crop, it will reproduce vegetatively. So vegetative propagation means through use of plant parts. So what is the next point? What is the role of apomixis? That is production of clonal seeds. What is meant by clonal selection? Next point, that is selection of superior clones. and what are the advantages of apomixis. So lecture number 16 will deal with mutation breeding in crop improvement. So in that, first we will see what is exactly mutation. So what is mutation? It is a sudden heritable change in genes. Then what are the different types of mutation? Micro and macro mutation. Then next we will see what are the mutagens, physical and chemical agents. Next we will see the induced mutation, that is artificial creation of variability through use of mutagens. Next point, selection, how to identify the useful mutants. And last, application of mutation breeding in development of improved varieties. So all these things we will cover in lecture number 16. That is mutation breeding in crop improvement. So in lecture number 17 we will see how to handle the segregating generation. So what is meant by segregating generation? So the generation in which we can see the maximum segregation. So as we all know, F2 is the most segregating generation in plant life cycle. F2 onwards, we can say like these are the segregating generations. So what are the different breeding methods? So for handling of segregating generation, there are three breeding methods: pedigree method, bulk method, and backcross method. So all these three we will see in detail. So lecture number 18 will deal with different hybridization techniques. So first we will see what is exactly hybridization, that is crossing of different genotypes. Then types of hybridization: intervarietal, interspecific, and intergeneric. Next point, what are the objectives of hybridization? So the most basic objective of hybridization is combination of desirable traits, right? Then next we will see emasculation and pollination. So what is emasculation? It is as simple as that, removal of anthers. And pollination means transfer of pollen from anther to stigma. So what is the role of this hybridization technique in creation of genetic variability? That also we will see. So in 19th lecture, we will see the variety release and notification. What is the need? What is the definition? That is official approval of new variety. Notification means. Then what are the testing stages? Multi-location trial, institutions like Central Variety Release Committee, State Variety Release Committee, and Indian Council of Agricultural Research. Notification means legal approval for cultivation. And last, distribution of seed. Okay. So in last, that is lecture number 20, we will see the intellectual property rights in agriculture. That is concept. First we will see concept of IPR. That is legal protection for innovation. What are the types of intellectual property rights? That is patents, trademarks, geographical indications, etc. Then we will see some institutions like TRIPS, that is International IP Agreements, WTO and WIPO, that is Global Regulatory Bodies. Then we will see plant variety protection. It safeguards the breeder's rights. And at last we will see the importance of IPR. So this is how we will complete the course from introduction to plant breeding till intellectual property rights in agriculture. So hope you will gain some valuable points, valuable information through this course. So hope to see you in detailed lectures. So till then, it's thank you very much. Thank you.